before we dive into some of the specifics of DNA replication in bacteria, I just want to do a quick overview of some basic concepts. So DNA replication is obviously very important for all organisms, and it's this very complex process involving dozens of proteins that ensure accuracy and that it's kind of done properly. And the two strands of DNA will basically separate, each serving as a template for synthesis of the complementary strand, meaning that the synthesis is semi-conservative, and each daughter cell or daughter chromosome will obtain one old and one new strand. And I'm going to go ahead and make a big assumption here that you're at least fairly comfortable with eukaryotic DNA replication. Hopefully you've heard it, maybe you've taken genetics. If not, here's a very, very brief overview. For eukaryotes, typically you'll have multiple chromosomes, and each individual chromosome is larger than the single circular bacterial chromosome for the most part. They are linear, so they have this chromosome end problem that's addressed with telomeres. Because they're so much larger, there will be multiple replicons or origins of replication where it can kind of be opened and replication forks can be created. You'll have what is called the origin replication complex that marks the origin of replication, and you'll have histones that are helping to pack the DNA into chromosomes. So that's just a very, very brief overview. If you need more of a refresher than that, I'll try to find some links where you can check it out. I don't plan on testing on eukaryotic DNA replication specifically. Alright, so the reason I'm mentioning eukaryotes at all, since I said I'm not planning on addressing that, is because there are some things that are shared among all three domains. So there is the semi-conservative replication that is universal for organisms, that you have to have one strand in order to make the other. Also, DNA synthesis is always 5' to 3'. You can only add things onto the 3' end of the DNA nucleotides, so that's why DNA and RNA synthesis is always 5' to 3'. You must have the template. Additionally, you must have an RNA primer because DNA polymerase, the enzyme that adds DNA, is incapable of just starting from nothing. So it's almost like a runway where you get prepared for DNA synthesis. You need an RNA primer to do that because RNA polymerase does not need a 3' overhang to begin adding nucleotides. You also need nucleotides, or DNTPs, deoxynucleotides. You will have a leading and a lagging strand. I'll get to that a little bit more, but hopefully that's familiar terminology. And there is a lot of replication machinery that's pretty much the same, although there are some differences in the logistics and how this takes place in bacteria and eukaryotes. One other really key distinction here is that in bacterial DNA replication, it can take place rapidly even while it's still duplicating. And so one thing that's interesting is that you can see the E. coli doubling time and the average rate of cell growth, and it actually takes longer to double the chromosome than it does for the cell to divide. And this is because during fast growth conditions, like in pure culture, you'll actually have divisions happening on a cell that's still being, or on a chromosome that's still being divided. So this can be very rapid, and there's no need for cell cycle checkpoints like what you have in eukaryotic cells. It just kind of happens. Alright, so since I wanted to talk about prokaryotic genomes, I just wanted to make a quick mention here about archaea, which are kind of like this mixture of eukaryotic and bacterial replication machinery. They are hard to classify. Sometimes they have things that are like bacteria, sometimes they have things that are like eukaryotes. We really don't have a good solid model for archaeal DNA replication, so we're not going to focus on that right now.